Okay, welcome, my dear friends. Very good evening to all the participants, young aspirants for CPGET 2025 and other competitive exams in plant sciences and plant biotechnology, and also other competitive exams like forest department, forest beat officer, forest section officer, and other competitive exams in the field of plant sciences, especially mycology. Dr. D.NAGARAJU sir is very eminent personality. He has written more than 10 books, intermediate and degree books, degree first year, second year, final year. The Telugu Academy books has written by Dr. Deshaboyana Nagaraj sir. Now sir is working in Government City College, Autonomous, Hyderabad, Nayapur, very nearer to the High Court of our Hyderabad. Sir is going to present today onwards in mycology, the topic mycology, the related to the other most important bits related to our CPGET and other competitive exams. So, my dear friends, listen carefully. Sir has huge experience in this field of mycology, especially fungal pathogens and plant diseases. Sir has very eminent personality. Telugu Academy books, degree first year, degree second year, and final year environmental studies, environmental science. Also, sir has written. So, in this regard, today is also sir is very busy with the other practical examinations. Even though the concern, the commitment of Dr. D. Nagaraj sir has given his valuable time for our young aspirants. So, sir has joined for our future. So, listen carefully during our Nagaraj sir. Class is very excellent. Definitely, I am sure you feel enjoyed. At the same time, sir has given his services from COVID pandemic situations to till today. Every year, Dr. D. Nagaraj sir has giving his valuable time for our students better rank. So, in this regard, we would like to invite Dr. Deshaboyana Nagaraj sir, Assistant Professor, Department of Botany, Government City College, Autonomous, Hyderabad. Sir, please welcome and share your screen. Start your session, sir. Dr. D. Nagaraj sir. Your voice is not audible, sir. Nagaraj sir.
The young budding botanist.
Good evening, sir.
Internet connection is unstable.
Nice to meet you, Nagaraj sir. You are looking very dynamic, even though from 7 o'clock morning to till now you are working with different levels of aspirants and students in Ambedkar University, and also we are ready to present to our young budding botanists better future. Please share your screen and start your session, sir.
Okay.
Yes, sir. We proceed, sir. Proceed.
Okay. Study about the fungi is called as mycology. You know, second most largest group of living biota. The second most largest group of living biota is fungi. The first one insect, you know, the study about the mites, that means moles and mushrooms, is called as mycology. You know who is the father of mycology? Micheli. Easy to remember. Micheli is the father of mycology. And you know, Anton Debori. Who is no Anton Debori? Who is that? is the father of modern mycology and also father of plant pathology. Disease caused by the so many fungi, bacteria, algae, whatever it is, plant pathology. Anton Debori is the father of plant pathology. That is very, very important. Who is the father of mycology? Who is the father of mycology? Here it is
Michael,
three.
sir.
You can see the Micheli. Michael is the father of mycology. And you also can see the father of Indian mycology, Butler. Butler, you know the Butler, E.J. Butler is the father of Indian mycology and also plant pathology. Father of mycology and plant pathology. These two are very, very important. Who is the father of Indian mycology or who is the father of Indian plant pathology? Who is that? E.J. Butler
Butler.
is the father of mycology or Indian mycology. Okay. And also father of modern mycology and plant pathology. Who is that worldwide? Anton Debori. Anton Debori is the father of modern mycology and also plant pathology. You know, in the competitive exam, what they asked, fungal plant body is. What is the fungal plant body? Fungal plant body is a tree. What it is? The fungal plant body is called as fundamental structure of the fungal body is hyphae. Hyphae. What it is? Hyphae. That means it is a filamentous like structure. All the fungi are look like a filamentous. You see, all the eukaryotes, all the fungi are filamentous. That filamentous type of the fungi. But some are there unicellular, single cell. Single cell organs are there, that is yeast and chytridium. That also asked in the competitive exam. Single cellular fungi, unicellular fungi are yeast and chytridium. You know importance of the yeast. Yeast, with the yeast only, you can able to run bakery and beverages, alcohol and bakery items. With the fermentation, you know, anaerobic respiration. With the anaerobic respiration, we could able to synthesize, we could able to produce alcohol, beer brand whiskey, and also bread, puff, cakes, but all we are being prepared with this fermentation. What happened? And also, you know, idli, dosha, vada, that also being prepared because of the fermentation by these yeast, fungi. Okay. And you know, if the single cellular organism, all the body will become the fertile part. All the body will be useful to form the spores and next generation. That we call as holocarpic. All the body. Holocarpic. All the body will become the fruiting body. Holocarpic. Okay. Whereas the filamentous form, some part will become it forms the spores, or also any fragmentation or budding. Various type of the reproductions are there that we will take in the next class. Various type of the reproduction: vegetative reproduction, asexual reproduction, and sexual reproduction. In the three ways. And you know the mycelium. What is the mycelium? Network of the hyphae. The so many hyphae, filamenta, these type of the filaments are interconnected, interwoven, and it forms the mycelium. Interconnected or interwoven. Thus, interconnected or interwoven hyphae is calling it as mycelium. The mycelium is all the plant body of the fungi. That's why we also call it as mycology. Mites, the molds. The study about this is called as mycology. Okay, mycelium and mycology. You know, in the mycelium hyphae, two types of the hyphae are present. That is septate and aseptate. You see the septate hyphae and aseptate hyphae, both. Aseptate hyphae also called as we also call it as what we call as
Spinocytes.
Cyno?
I think has
Any?
sir.
There no students are there. Okay, cynocytic. There is no septa. That's why more number of the nucleus are there. That we call as cynocytic. Whereas in the septate, each cell having the single nucleus. The septate and aseptate. And you know, based upon the septa, also again 3 types are there: simple septa and simple porous septa. Where it is? This one. Okay. Simple septate are there and simple porous septa are there and porous septa. You see, septate porous. Simple porous septa is present in. There's so many competitive exams they ask. Simple porous septa. You see here, this one, they having the simple lineage, simple space, empty space. With that, it will be interchange all the cytoplasm and nucleus material from one to other, all the body. The simple porous septa. And whereas in the basidiomycotina, you can see the pore septa. It having the paranthrozomes, 2 paranthrozomes. As like a copper chipper, laga, up and laga. You see, up and down. These paranthrozomes are present. These paranthrozomes having the gap in between this. Where the porous space is there, that porous space, 2 sides septa will be bulged, swollen, and it forms the cup-shaped structures in both the sides. All the structure is called as pore septa. These pore septa are present in Basidiomycetes fungi. You know Basidiomycetes fungi? Actually, we will study the classification in next. Classification. Fungi are classified into majorly 2 groups. What are those 2 groups?
Mycota.
Mycota and Mycota and Myxomycota.
Myxomycota. Okay.
False fungi and true fungi. Divided into 2. False fungi and true fungi. Okay. Myxomycota we also call as slime molds. Jiguru slime molds. It having the water drop structure. Mold, it having the water drop, very slimy and water drop like structures. That are slime molds. That are paramecium like structure. There is no specific structures are there. Plasmodium, also called as the plasmodial like fruiting body in the Myxomycetes. And the Eumycota, you know, again, true fungi are again divided into, what are those? Do you have any idea? Masticomycotina, Zygomycotina, Ascomycotina, Basidiomycotina, and Deuteromycotina. You know, these are the 5. You know, in the competitive exam, they ask, which among is the flagellated fungi? Which among is the motile? Which group of fungi are motile? Which group of fungi are flagellated? Which group? Only the one, that is Masticomycotina. These are majorly look like algae. Similarities, having the more similarities with algae. That's why we also call it Phycomycetes. Phycomycetes. Phyco means, Phyx means algae. Phyx means algae. Myce means fungi. Fungi which are look like a algae. That's why we call it Phycomycetes. Phyx means algae. Study about the Phyx is called as phycology. You know who is the father of phycology? Fritsch. Fritsch classified algae into 11 classes. And the father of mycology is Micheli. Algae are autotrophic, chlorophyllous autotrophic thalapides. Fungi are echlorophyllous heterotrophic thalapides. That is the difference between the algae and fungi. Okay. You know, so many people, they couldn't able to give the definition of the algae and fungi. What is the definition of the algae? Chlorophyllus
Algae is
or
a photosynthesis.
algae. Achlorophyllus, heterotrophic thalapids are fungi. You know, plants are divided into two. What are those? Cryptogams and phanerogams. Cryptogams and phanerogams. Non-flowering plants and flowering plants. Harisha, non-flowering plants and flowering plants. Cryptogams and phanerogams. Those are not having any flower. That all are cryptogams. The cryptogams again divided into how many types?
Three types.
Three types.
What are those?
Thallophyta, bryophyta, and pteridophyta.
Yes, Thallophyta, bryophyta, and pteridophyta. Thallophyta. That means Thallophyta means plants which having thallus-like plant body. There is no root, stem, leaf modification. There is no specific root, stem, leaf-like structure. These all look like a mat. Only unique type of the structure, filamentous structure or unicellular structure. That are thallophytes. Thallophytes again divided into two. What are those? Algae. Algae are chlorophyllus autotrophic thallophytes. Fungi are achlorophyllus heterotrophic thallophytes. Both are thallophytes. Both are having the thallus-like plant body. Both are look alike, but due to chlorophyll, algae are in green and different color because of the different chlorophyll pigments, red, brown, golden color. Jewels of the plants? Jewels. Jewelry, jewels, ornaments of the plants. That are very attractive. Ornaments. Then what you are wearing the ornaments. The natural ornaments.
Diatoms.
Diatoms. Bacillariophyceae algae are called as jewels of the plants. Okay. Then achlorophyllus heterotrophic thallophytes are fungi. Studying about this are called as mycology. Fungal cell wall is made up of it. So many competitive exams they ask the question. Fungal cell wall is made up of it. Usually all the plant cell wall is made up of it. All the plants cell wall is made up of it.
Cellulose.
But whereas in the fungi cell wall is made up of it.
Chitin.
Chitin. Fungal cell wall is made up of it. Chitin. And one more thing, the majorly chitin. Chitin we also call it as fungal cellulose. But in the higher fungi, whereas in the Ascomycotina, along with the chitin, glucans are present. What are these? So many competitive exam I asked. The Ascomycotina cell wall is made up of it. Chitin plus cellulose, chitin plus glucan, chitin plus mannan, like that they ask the question. Along with the chitin, glucans are present in Ascomycotina, and chitin plus mannans are there in the Basidiomycotina. And in the whereas in the oomycetes, that means in the Mastigomycotina, along with the chitin, cellulose also present as like a plant cell wall. Chitin and cellulose. These are the differences. They ask the question so many times. They ask the question. Suppose lower fungi cell wall is made up of it, chitin and cellulose. In the higher fungi cell wall is made up of it, along with the chitin, glucan and mannans are present. Okay. Cell walls are very, very important. Ascomycotina cell wall, along with the chitin, glucans are present. Whereas in the Basidiomycotina, chitin and mannans are present. The fungal stain, fungi stain with lactophenol. We can observe clearly by staining with the lactophenol blue. By staining with the lactophenol blue, we can see the fungi very clearly. That also question how many times fungal stain. Fungal stain is lactophenol blue. Okay, that lactophenol are observed by the chitin so that it will be observe the blue color, lactophenol blue color. Okay, and you know, some of the fungi, it shows the dimorphism. Dimorphism means it appears as like the both, as like a mole, single cell organism, or as like a filamentous, the both. Sometimes it may be as a saprophytes, and sometimes it may be as a parasite. The both the habitat, both various type of the nutrition type, various type of the structure it having. That we call as dimorphic fungi. It shows dimorphism. Best example is Histoplasma capsulatum. Okay, and you know, homokaryotic and heterokaryotic. That means what does it mean, homokaryotic and heterokaryotic? If all the nucleus are in same, that is homokaryotic. If the mycelium contains a different type of the genetic material, nucleon, that we call as heterokaryote. Heterokaryote present in where? In which stage? Where it is present? You know, in the secondary mycelium of the basidiomycetes. You know, after clamp connections, outer ring road, bypass. Bypass roads are formed in the higher fungi, basidiomycetes. That bypass road is called as clamp connection. They connect with the one cell to other by ring, outer ring road like structure from one cell to other. With that clamp connection, cells are nucleus are interchanged. So that heterokaryote, different type of the nucleus will be formed. You know, multinucleate, that we already know, we already told you. If there is no septa, multinucleus are present. If there is no septa, cenocytic. Okay, that we call it cenocytic. And also you can see, already told you the difference between the algae and fungi. Algae mostly aquatic, fungi mostly terrestrial. Algae having the chlorophyll, fungi having the no chlorophyll. Okay, and algae are autotrophic, fungi are heterotrophic. You know, majority of the algae are eukaryote, but only one. You know the which one is the prokaryotic algae? That also asked in the question so many times. Prokaryotic algae.
Cyanobacteria.
Cyanobacteria, blue-green algae. Why we calling a cyanobacteria? Why blue-green algae? We are calling it a cyanobacteria. Why? Why they included in the bacteria?
They have bacteria character.
Bacteria?
Characters, sir.
What are
Prokaryotic organism. Prokaryotic organism.
Prokaryotic structure. What are those? Nuclei. There is no specific nucleus, nucleolus, nuclear membrane. Nucleids only present. Yes, and cell wall. Cell wall also made up with mucopeptides, not the cellulose. These two different character, distinguishing character. Okay. Due to that, we also studied in the bacteria as a cyanobacteria, blue-green bacteria. Okay, cyanobacteria. Okay, even the reserve food material also somewhat change as like a glycogen, cyanophysin starch. Okay. And then what do you study? Okay. You know the hyphae. What is the basic fundamental structure of the fungi?
Hyphae.
What is the basic fundamental structure of the fungi? Hyphae.
Hyphae.
Okay. That hyphae modifications are there. Not all the hyphae are same. Not all the hyphae are unique. You know the hyphal structure, if it is a loosely coiled, loose, very loosely as like a tube, separate tube like structure, that we call it as pseudoparenchyma or prosenchyma. Prosenchyma or pseudoparenchyma. If it is tightly coiled without distinguishing, separate, we can't separate with without damage. Without tightly coiled, interwoven, that is plectenchyma. Plectenchyma. Prosenchyma and plectenchyma. If it is loosely arranged, hyper loosely arranged, that is prosenchyma or pseudoparenchyma. And if it is tightly arranged, that is plectenchyma. And sometimes the fungal hyphae is also look like a root shape structures that we call as rhizoma, root-like structure. Rhizome means root, morphologically, structurally, root-like structure. That we call as rhizoma. Rhizomas also present. You know, Armillaria milia. You know, Armillaria milia, there's so many pictures you have seen, I think, in the forest while they are going in the forest, it's shown the light, it's shown the way. As like a miniguru purugu. It gave the light, it eliminated the light, so that it gave the light, yellow color lights. Armillaria milia. Okay. And you know the sclerosium. Some of the hyphae are modified into, you know, the sclerosium. Sclerosium means immediately you have to remember what it is. What the sclerosium? Where in the sclerosium you have studied? Claviceps purpurea, ergot of ray. Ergot of the ray disease caused by Claviceps purpurea. That fungi from the sclerosium. That means hard hypal structure. We're having the hard structure. Okay. And you know, Apressoria and Hasturias. Apressorias and Asturias. Apressoria. You know, Apressoria and Asturias, you can see the structure here so that you have an idea. You see here, what are the, you see, rhizoma, root-like structure. These are root-like structure that we call a rhizoma. And it having the sclerosias. You see, Claviceps purpurea. It having the sclerosias. And here you can see the Apressoria. The tube-like structure with extra tube-like structure of the thallus, hyphae, which are enter inside at the tip of the hyphae. That we call as Apressoria to absorb the nutrients. And whereas Asturias, these are finger-like projections, these enter into the host to absorb the nutrition from the host. Especially in the parasitic fungi, the Asturias are present. You know, Asturias, 5 types of the Asturias are there. Okay, intercellular and intracellular, and what are those? Vascular, arbuscular, and multitubular. The various structures, 5 types of the structure, intra and inter. Intra, that means it enter into the cells and in between the cells to absorb the nutrition from the host. Especially in the parasitic fungi, Asturias are present. Asturias are useful to absorb the nutrition from the host. You know, based upon the nutrition, fungi are divided into how many? How many? You know, fungi are what type of the nutrition it shows? Fungi?
Heterotrophic.
Heterotrophic. Heterotrophic nutrition is divided into 3. What are those? Saprophytes, parasite, symbiont. Saprophytes, parasite, and symbiont. Okay. Saprophytes means which grown on dead organic matter, dead plant or animal. These are decomposers, which decompose the dead organic matter. Natural scavengers. You know, the bacteria and fungi are natural scavengers, which clean the environment, which clean the debris and dead bodies from the earth. It make it fertile soil, highly fertile soil by decomposing this dead organic matter. Otherwise, we can't stay even upon the 1 kilometer upon the land. If all the dead bodies still remain, there is no decomposition. If there is no saprophyte, there is no natural scavenger. We have no space even to stand upon the earth. Trillions of the living organisms are died. If are all accumulated, no space. If there is no decomposers. Okay. I should salute to the decomposers. Otherwise, we can't. Even any organism if it died, we can't inhale that smell. At least 3 to 1 week, 3 days to 1 week, that smell will come. If the dead bodies will remain, we can't inhale. Okay. That all making clean the environment and earth by these saprophytes. You know, the saprophytes, mucor, rhizopas, penicillium, aspergillus, and fusarium, but it also act as a parasite whenever the optimal environmental condition, specific hosts are present. Okay, and then parasites. That means we take the food from others. It's totally depend on the host. It may kill the host. It may kill the host also. That are parasite. You know the parasites again 2 types: obligate parasite and facultative parasite. This question also so many times asked. Which among is the obligate parasite? Puccinia is the obligate parasite or facultative parasite? You know Puccinia major question. We trust. You know Puccinia, we trust. We trust disease. Macrocyclic life cycle. You have studied. 5 stages are there. 2 hosts are present. 2 hosts. What are those 2 hosts? Puccinia gramineous decide 2 hosts. What are those 2 hosts? So many times they ask the question. Puccinia gramineous the primary host is wheat, godma. And the secondary host is barberry plant, weed plant it is. That is the secondary host. It carries the spores to complete all the life cycle. You know, K.C. Mehta has done the study on the rust. He identified the Puccinia spores are coming from the north to south, south to east with the monsoons. That's why they stopped the cultivation of the wheat in the southern some years together to stop the dispersal of the spores from south to north, north to south by northeast monsoons and southwest monsoons. Okay, different time is there for the cultivation of the wheat in the north and south. That's why whenever it gets mature, the spores in the north that are coming to the south by the southwest monsoons. And then in the northeast monsoons, they are going from here to there. With that uridospores causing the disease to the host of the whole area. Okay. You know the 5 types of the spores in the Puccinia? What are those 5 types of the spores? Can anybody? Harsha, Jaya? Uridospore, Kilitospore, Ichthyospores, Pycniospores, Basidiospores. You know what type of the spores? Which spores are there on the host plant, primary host? So many complete exam they ask. Uridospores and Tildospores are present on wheat. And Pycniospores, Ichthyospores are present in barberry. You know, Basidiospores are only on the soil, not in any host. Okay. And then facultative parasites means, all of you, are you listening? Facultative parasite means, usually these are saprophytes. Is it audible? Usually these are saprophytes. But whenever the specific hosts are present, it will be turned into as a parasite. I already told you, Fusarium, Pythium. Fusarium and Pythium. These are actually saprophytes. But whenever the specific plants are there, it causes the wilt. Wilt disease. You know the wilt disease? Wilt disease of so many crops, especially in the banana. And you know Fusarium wilt and Pythium. Pythium Phytophthora. You know the Pythium and Phytophthora. This comes under Muscomycotina. Okay. And then the facultative saprophyte. That means usually these are parasites. But if there is no specific host present, in the absence of the living host, it will become the saprophyte. Dash అయితే గుర్రం గడ్డి తింటది అంటారు కదా, like that. Actually, it is a parasite. But if there is no host, it will become the saprophyte to survive. It decomposes the dead organic matter, it absorbs the nutrition. These are the basic types. You know, the so many competitive exams they asked obligate parasite and facultative parasite. Okay? Obligate parasites means the totally parasite. If there is no host, they can't survive without the host. That are obligate parasites. Facultative parasite means this can survive without the host sometime as a saprophyte or as a symbiont. Facultative saprophyte means which also can survive as a parasite if there is as a saprophyte if there is no host. Okay. And the major one, fungi, another type of the nutrition is symbiont. You know, two types of the symbiont which are there with the fungi. What are those? Lichens and mycorrhiza. Lichens and mycorrhiza. You know what are the lichens? Lichens means what are the lichens? Lichen is a single organism or multi-organism? What it is?
Two organisms.
What are those?
Fungi and algae.
Algae and fungi. Both are there. Okay. Algae and fungi. Both are symbiotic association of algae and fungi. It's a lichens. The symbiotic association. Algae synthesize the food. Fungi, it gives the shelter to the algae. You know, this one, we also call it as idealistic marriage. Idealistic marriage between the algae and fungi. Couples. Idealistic marriage. The lichens having the idealistic marriage in between the algae and fungi. You know, king and slavery mechanism also present in the lichens. You know, fungi act as a king. Algae act as a slavery to produce the food, to synthesize the food, to prepare the food for the algae, for the fungi. Algae, they don't reproduce, they don't do the reproduction in the lichens, but only fungi do the reproduction. Fungi only form the spores, fungi only disperse, fungi only multiply their generations. Okay, you know, actually, Scribbly and Metlin, Scribbly, the scientist he identified two fungi and one algae. Trinity in the algae, or trinity in the lichens, not the two. Three organisms, two fungi, one algae. Two fungi, one algae are present in the lichens. Okay, that are observed, identified by the staining technique. With the stain, it has shown three different stains, so that they observe three different organisms. One major is the yeast basic aspects of the lichens. That's why we can't generate the lichens artificially by mixing the algae and fungi. Okay. And you know the lichens, we also call it as lichens, we also call it as
Symbiotic association.
symbiotic association, and we also call it as, you want our normal name,
Solution indicators.
stone flowers, and you know Silaja, Shila means stone. Cha means living upon the stones. Okay. You know, lichen we will take the another class. Okay. One class for the lichen. Lichen also 1 or 2 questions will come from the lichen also. And the mycorrhiza, you know the mycorrhiza. Myco means fungi, rhiza means roots. Fungi which are associated with the roots of higher plants, that are mycorrhiza. You know, the mycorrhizal network is the largest network than our telephonic network or any wireless network, any network. You know, mycorrhiza widespread in the soil. It penetrates, percolate into the soil to the wide distance. The mycorrhiza is present, widely present. This calls for the supplying of the nutrition to all the land plants. The mycorrhiza, if it's more mycorrhiza, plenty of the mycorrhiza is there in the soil will become very fertile. But due to utilization of the pesticides and more and more pesticides and fertilizers, we are killing the mycorrhiza. We are making our soil will become nirjivam, sessile, sterile soil. We made already our soil sterile soil instead of fertile soil. If the fungi, mycorrhiza is present in the soil, that will be very fertile soil. It gives the more fertility, more fertile, more crop, and high yielding will come. You know the mycorrhiza types are there now? Do you know the mycorrhiza type? Mycorrhiza.
In cycles plants.
7 types are there, the mycorrhiza. Ectomycorrhiza, endomycorrhiza, ectendomycorrhiza, warm, vascular, arbuscular mycorrhiza, corticoid mycorrhiza, ericoid mycorrhiza, like this, archic mycorrhiza, like this. 7 types of the mycorrhizas are there. Okay, major mycorrhizas, arbuscular mycorrhiza, warm, which form the vesicles and arbuscles, and it absorbs the nutrition and supplies to the plant. Okay, vascular, arbuscular mycorrhiza. Major vascular, arbuscular mycorrhiza is glomus and hylococcus. Both are there. You know, the another type of the nutrition in the fungi, that also very, very important. Predaceous fungi. Predaceous means which survive on the other organisms, other animals. Predaceous. It kills, you know, there's so many insects, mites, thrips, it having the moles upon it. It kills that insects, mites, thrips. You know, nematophagous fungi also there, and insectivorous fungi are there. Nematophagous fungi and insectivorous fungi, these all are called as predaceous fungi. They survive on other animals which kill that organisms. Not only on the plant, these also parasitize on animals, that are predaceous fungi. Nematophagous fungi, entomogenous fungi, and insecticidal fungi are there. You know, Nematoptera, that is nematophagous fungi. Verticillium, Artemisium, these all are insectivorous fungi. It kills the insects. Okay. Can we stop? We will discuss the reproduction and classification tomorrow. Huh?
Okay.
What we have studied at? We have studied about the fungi. Study about the fungi is called as mycology. Fungal body is called as hyphae. Plural is hypa. Singular is hyphae. Singular is hyphae. Interconnection of the hyphae is called as mycelium. You know, fungi are heterotrophic. You know, Puccinia is a facultative parasite. Only survive on the host. And saprophytes are Fusarium, Pediptera, Pythium are saprophytic fungi. You know, flagellated fungi, motile fungi are masticomycotinous fungi. Okay. And the lower fungi are masticomycotina, zygomycotina. You know, the zygomycotina, mucormycosis at the time of corona. It attacked the sugar patients who having the corona. It caused for the killing of the corona patients also. It damage to the eyes and skin.
Black fungus.
So many are died after curing from the corona also because of infection of mucormycosis. Mycosis means disease caused by the mites, fungi. Okay, and the father of mycology is Micheli. Father of modern mycology is Anton Dibori. Okay, and the fungi having the holocarpic and eucarpic type. Holocarpic means all the plant body will become the petile pod, that is yeast and chytridium. Which among it is the holocarpic fungi? Yeast and chytridium. Okay, which is the cenocytic fungi? That means zygomastous fungi. Mucor, rhizopus, these are cenocytic fungi. They are having the multinucleate hyphae. Multinucleate hyphae is present in the rhizopus, mucor, obsidia, and all the mucorales. Okay, multinucleate. Okay, and the simple porous septa is present in Ascomycotina. Dallipore septa is present in Basidiomycotina. These are the very, very important points which we have discussed. And in the next class, we will discuss the reproduction and classification. And in the next class, we will discuss the economic importance of fungi. And in the next class, we will discuss the lichens. Okay? Okay, then.
Thank you, sir.
Okay, thank you. Thank you all. We will go through this. We're having the very, very important points. Second most largest group of the living organism is the fungi. Okay. Yeast. Without the yeast, there is no bakery beverages. Fermentation, anaerobic respiration. That giving us to trillion dollars of the money. So many are surviving with the bakery and beverages. So many are died. So many are injured with the beverages also. Okay. All right.
Okay, sir. Am I audible?
Yes, sir.
Okay, thank you very much, sir. Today, we're going to wind up the session. Nagaraj sir, your voice is not audible. Okay. Tomorrow we will meet. We will going to wind up the session. Thank you very much, sir.